very good morning to Dr. Azuraidi. We are from Group 2 and today we will be presenting on base excision repair. So before that, we will be introducing ourselves. My name is Nancy Nomia, metric number 210358.
My name is Iman Ibrahim Hamzah, metric number 210247.
My name is Muhammad Arieff bin FAKHRUR RAZI. My matrix number is 21022350.
My name is UMAIRAH FARISAH and my matrix number is 210177.
My name is NURUL AFIQAH BINTI NORAZIRUDDIN and my matrix number is 210213.
And my name is HANISAH BINTI ABDUL. My matrix number is 210163.
Thank you, everyone. Unlike other processes such as RNA and protein synthesis, which occur many times, DNA replication occurs only once. Therefore, it is essential that the fidelity of DNA replication to be as high as possible to prevent mutation. However, besides UV radiation, nitrogenous base of DNA can be damaged by mechanisms such as alkylation, deamination, and oxidation. The presence of these modified bases results in incorrect base pairing and mutation in DNA. Mutations are frequently harmful, even lethal to organisms. So nature has devised several ways to ensure that the base sequence of DNA is copied faithfully. One of the critical protective systems is base excision repair that mitigates environmentally induced DNA damage, repairs small non-helix distorting lesions, and abasic sites. Now, let's take a look at the mechanism. The enzyme involved in this mechanism is DNA glycosylase. DNA glycosylase identifies the modified base and removes them out of the double helix. The empty region in the DNA where the base has been removed is known as the AP site. AP site stands for abasic site, which means it is devoid of any purine or pyrimidine site. Sometimes it is also referred to as apurinic or apyrimidinic site. In the next step, AP endonuclease cleaves this region to yield free 3' OH and 5' phosphate. DNA polymerase I then identifies the 3' OH and adds correct bases in 5' to 3' direction. During this step, DNA polymerase I also carries out 5' to 3' exonuclease activity to remove the old bases. So once the activity of the DNA polymerase I is over, the remaining gap is sealed by DNA ligase. So that is all about the mechanism. So what are the properties that is able to distinguish base excision repair from nucleotide excision repair and mismatch repair is what kind of bases are being removed here? Only chemically modified bases. Secondly, how the bases are being removed. DNA glycosylase remove the bases by breaking the glycosidic bond. So this is all from me. I'll pass it to the next presenter.
Okay, thank you, Nancy. Now I will present about the pathways in base excision repairs. There are two pathways in for base excision repairs. The first one is the short patch repair. The short patch repair is when the cell undergoes mutation, glycosylase cleaves the base and flips the double helix, thus leaving the AB sites. Then AP endonuclease and SRCC1 cut the 5' of the AB site. Then exonuclease comes in to cut the 3' of the AB site. DNA polymerase beta fills the gap with the correct base pairing using undamaged template. It also leaves behind 5' and 3' nick. As SRCC1 and ligase repair the nick. During ligation, which is what I just explained just now, ATP is converted to AMP. Next, I pass to next presenter.
Okay, thank you. Right now, I'm going to talk about long patch pathway of BER. Long patch repair, instead of repair small one, it repairs a patch several nucleotides long. In order to achieve this, a different DNA polymerase, which is delta epsilon, adds several nucleotides that displace the original nucleotides. This results an overhang of oligonucleotides called a flag, which contain the damaged base. In presence of replication factor called PCNA and FEN, this remove this flag before a DNA ligase cells terminate. The mechanism of long patch DER is particularly useful to repair damage resulting from ionizing radiation. Next, I will pass to the next presenter.
Thank you, Arif. So, every reaction needs an enzyme to lower down activation energy.
Yes, correct, Aiman. Same goes to base excision repair.
So, we need a group of enzymes called glycosylases. Each glycosylase detects and removes a specific kind of damaged base.
For example, a chemical reaction called deamination can convert a cytosine base into uracil, a base typically found in RNA. Okay, I'll pass to the next presenter, HANISAH.
Okay, thank you everyone. So let me briefly talk about this process. What has everyone explained before. So enzyme called DNA glycosylase extracts the DNA damaged part, and then an enzyme called AP endonuclease finds the gap in the sugar phosphate backbone and remove neighboring nucleotides. And next, DNA polymerase assembles the missing base, and finally DNA ligase rejoins the sugar phosphate backbone. So in conclusion, base excision repair or BER is very important for our body. This is because BER is a key genome maintenance pathway that removes endogenously damaged DNA bases that arise in cell at very high levels on a daily basis. It is needed to correct the DNA damage from oxidation, deamination, and also alkylation. Failure to remove and repair these damaged DNA bases lead to increased levels of mutagenesis and also chromosomal instability, which then have the potential to drive carcinogenesis. Some diseases caused by defect-based excision repair that I got from the internet are neurological disorder and also cancer. For example, Alzheimer's diseases and colon cancer. Functional BER is required to prevent aging. In short, it is proven that BER is crucial in protecting us against cancer and neurodegeneration and also aging. So that is all from us. Thank you.